Thank you for inviting me. It's a pleasure to be here. My first trip since COVID, and to be with all of you. So I'm going to talk about individual differences, gene-environment interplay, and epigenetics. I just want to acknowledge some of the funding sources, and I'm also going to talk a lot about fruit flies. And by the end, you might find them really interesting. So individuals of the same species display remarkable variation in behavior, even in identical contexts. But the molecular mechanisms that underlie this variation is poorly understood. When I was an undergrad student, I was asked to do a project in a developmental course, developmental genetics, and I knew I liked behavior and bothered the professor and said, could I look at them in an open field and see what they're doing? And she said, you can never do genetics in behavior because behavior is not a phenotype. That would show you how long ago that was. But I was very persistent and took the maggots from different bottles and went to orchards, and I developed a little open field assay for fruit fly larvae, put them in for 5 minutes on a yeast-covered petri dish. How do I get in? Is that an arrow that comes here? Yeah?
Arrow, yeah.
I don't actually know where it's going. Okay, doesn't matter. So, whoop. So you can see on top that there's a yeast-covered petri dish. I put a larva in the middle for 5 minutes, and some of the animals moved a lot. I called them rovers while they ate, and others moved a little bit. I call them sitters.
This is going to go bigger.
Okay. So how do I know? Oh, now I see everything. Yay. Okay. And it's not that the sitter was a couch potato. If you put them in the absence of food, they both move the same. So those were the original phenotypes. And in natural populations, we saw both types. We saw if you took larvae from a single fruit, 30% were what I call sitters and 70% were rovers. And also, the sitter would move from, if you put it not on food, it would move to the nearest patch and stay there, a group of them, and the rover would move from patch to patch. And so we did a lot of genetic analysis to try and understand what genes contribute to these rover-sitter differences in behavior, and found many genes involved using QTL analysis, which is kind of, it's like GWAS. But there was one gene that had a more major effect, and that was the gene that we went after. We mapped it using recombination mapping, deletion analysis, and eventually cloned it, and that's the gene that I'm going to talk to you about today. There are other genes on other chromosomes that we have also studied, but the effect size of those genes are smaller, and this was happening in the late 1970s. So you can imagine how difficult it was to find genes that influence natural differences in variation. We weren't making a mutant and having a wild type that's homozygous and altering one gene and then finding it. In the gene, there may be 200 SNPs, and we were trying to figure out which gene is important and which SNPs might be important to that contribution. So it was, at the time, people were very interested in it. Because my background was in evolution and ecology, I also wondered, why are there two types? Why isn't it that one animal can't just do everything? And why do we have these two distributions? And we, in those days, did experiments on density-dependent selection. Under very high densities, you select for the rover type and the genetic variant. And under low densities, when it's not crowded, they don't waste energy in locomotion, and you get more sitters. And you can grow them together and show that when you're rare, when you're the uncommon type, you have a higher fitness, you get more progeny. So it's a mechanism to maintain polymorphism. But today I want to talk to you about all the different things we've learned because of this gene. So when we cloned the gene, we found out that it was a cGMP-dependent protein kinase, which is a signaling molecule, and it wasn't so well known in those days. It phosphorylates a lot of different proteins, and upstream NO activates cGMP and affects this kinase. So it encodes for that molecule, and I'm just going to call it PKG. And individuals have different alleles, different forms of the gene, rover and sitter, and both of those alleles give you predispositions to behave, to move more or less in a feeding environment, but there's context-dependent plasticity. So you can food deprive a rover and turn it into a sitter. And I'm going to show you different kinds of plasticity. Another kind of plasticity, you can vary food deprivation, make it chronic in early life, see what happens later in life as an adult, and make it acute. We've done a lot of experiments like that. And 4 is responsive to the abiotic and social environment. I like to think that the genes are listening to the environment. It's not just a one-way process, but there's gene-environment interactions, and there's an interplay between the two. So the other thing is that foraging can act on different time scales, and it has a function in the synapse, for instance, and this is a very quick time scale where it affects neurotransmission, and its function, there's multiple functions at the synapse, and there's pleiotropic functions. So it influences neurotransmission in glial cells by extending nerve terminal growth. And in presynaptic cells, it influences vesicle recycling. And so you can see even at that small level that it's having in seconds a synaptic phenotype. And also there's changes in gene expression, which I'm going to talk about, that influence the phenotypes, and we'll get to epigenetic modification and how it's regulated. And after identifying this simple phenotype, we realize it's highly pleiotropic and affects many things. It affects food intake, fat, and food-related movement, so a suite of metabolic traits, and sucrose responsiveness, response to pain, learning and memory, stress, sleep, social behavior. And so we wanted to understand, how is this one gene accomplishing all these pleiotropic phenotypes? And a lot of the talk will be about that today. Okay, and the study on epigenetic processes, which was part of Ina's PhD thesis, will show you an example of genetic by epigenetic interactions in producing the Rover-Sitter differences. So that's just a little highlight. So foraging is conserved. We find it in almost everything that you look in. It's been studied a lot in social insects. So for honeybees and ants, the young nurse that looks after the babies has very low levels of foraging. And then when it goes to forage, the foraging gene expression increases. And so in that case, it's within the lifetime of the individual fly that the expression, the individual honeybee and ant, that the expression changes, where in C. elegans, the worm, and flies, we have these allelic variation. And we see lots of SNPs in the human foraging gene. I'll talk a little bit about that, and a little bit about mouse models. And so almost whatever organism you pick, you find this gene. The protein is highly conserved, the sequence that makes the protein. So it's interesting. For fun, I want to tell you one story about the human gene. And it's, you of course all know what a single nucleotide polymorphism is, SNP. And this was a project I got together with a social psychologist and a neuropsychologist, because I gave a talk and someone said, we are sure that we have rovers and sitters in humans. And I thought, that's really funny. And they told me that the people who measure personality traits, they call individuals locomotors or assessors. And they have a 12-point questionnaire where they ask the person questions, and they get a score on this, on a locomotor scale. How much do you tend to be locomotor? And then on an assessor scale, how much are you more assessor? So locomotor is a person that's always on the move. And I don't just mean physically. Your mind is always on the move. You go from one thing to the other. The assessor is the individual who sits there, collects all the information, weighs the pros and cons, and then eventually makes a decision. It's like the difference between my husband and I if we have to buy a television. You know, he does all the research. He used to be a dentist. All the research, 4 or 5 months later, he gets the best television, and I would have decided in 2 minutes, right? And interestingly, in business, psychologists help to have locomotors and assessors work together in certain proportions that are like the same as the rover-sitter types in the flies. And you obviously can't have a locomotor leading a group of assessors or vice versa, or it's going to drive everyone crazy. Anyways, so we took a SNP in the foraging gene that is in the 3' UTR. It was a candidate gene study. And we knew that there was differences in expression according to the SNP in the dorsolateral prefrontal cortex. And this has been replicated 3 times in 3 independent samples. now, and we are trying to figure out how to do a GWAS study on this, but the phenotype is particular. Like, I don't think they measure that in many GWAS studies. But anyways, so on the locomotor and assessor scale on the y-axis, you see that there's significant differences. So those with the AA genotype tend to be more assessors, and then the CC more locomotors with the heterozygotes in the middle. And then the psychologists set up a foraging assay in the lab. And so they didn't actually put food out. They had a screen, a computer screen, and little berries dropped from different corners, and the student sitting there had to touch the screen, and the computer was figuring out how many berries were got in a period of time, and they did various other things like make big berries, small berries, etc. And they noticed that humans have different styles of foraging. So for the humans, us, right? I work on flies, so I always say humans to distinguish. So anyways, there's a lot of individuals that use a boundary bias. They go around the edges, and then a berry comes, and with their eyes, and they grab the berry. And other individuals were more systematic, looking at the screen like this when their eyes were being tracked and grab a berry, and some were just mixed. They were kind of all over the place. So when we look at our rover and sitter flies, and I'm going to talk to you more about the epigenetic regulation of these behaviors, if you put a fly in an arena, and there's 4 drops of food in that arena, here, anyways, it doesn't matter, here and here, what the sitter does, the fly adult, is it hugs the boundaries. And then it will make a quick dash in the middle to get some food and come back. But most of the time, it's hugging the boundary. And the rovers spend their time going into the middle a lot. And we used to put jumping spiders in the middle of the assay to try and figure out, what the risks and benefits are of doing this. But anyways, when we looked at the human data, it was pretty interesting because the boundary bias type looked a lot like the sitter, and the rover was probably mixed, maybe systematic, hard to say. But we could look at the genetics also, and when we did that, we saw that the boundary bias was the people at AA were heavily in favor of that style. So as I said, we've done this now 3 different times in about 300 or 400 students each time, and we see this kind of pattern. So, I don't know if it's real, but it's a great, fun, and interesting story. It needs to be done in bigger cohorts somehow. Okay, so pleiotropy, that's a gene having multiple functions, and I want to talk to you about that. And the question is, how does D. melanogaster, the fruit fly, the gene foraging, accomplish its multiple functions? And we investigated that by looking at gene structure and gene regulation. So when we look at this gene, it's pretty complicated. Like many human genes, it's 40 kb, and this is the genomic sequence up here. And these are all the transcripts. There's 21 transcripts. This is the 3' end, the kinase domain, shared by all the transcripts. There's 2 cGMP binding domains, and then the 5' end differs. And Aaron Allen, who is a PhD student in my lab, is now a postdoc at Oxford. He experimentally identified 4 different promoters. And so these promoters direct a subset of transcripts. So promoter 1 directs a subset of these transcripts to certain tissues at certain times. So we see a modular structure is involved. And then similarly, promoter 4, I'm looking for my little mouse, but anyways, you can see promoter 4 directs these transcripts to a subset of tissues, which we found out about. So what Aaron thought he would do, first of all, is make a null or delete the entire gene. This was tricky. It was before CRISPR, so he used something called homologous recombination, and he deleted the entire gene, and so that was the null, and then he put the whole piece back into the fly. So we had the gene missing and the gene put back. And we were able to use those tools to do a dosage study. What happens to movement patterns, path length, how much they eat, and their fat levels as a larva with the absence of the gene, with 2 copies of the gene, and with 4 copies of the gene. And so just to show you that this worked on Westerns, that's a null. It has no protein. And then here's the RNA levels. The null is here. 2 copies and then 4 copies. And so when we look at these 3 phenotypes, the path length, how much they move in a dish, food intake, how much they eat, and fat levels, triglyceride levels, we see an indication that there might be differences in the regulation of these three phenotypes because of the direction that the change was in. And also a suggestion that maybe there's tissue or developmental specific timing involved in this. And for fat levels, the more of this gene we put in, the thinner, the leaner the fly got. And the same is seen in mouse models for this gene. And in human GWAS, it's been shown to be involved in obesity. So here's a mouse that has a transgenic copy of the human foraging gene, PRKG1. This is a normal mouse, it's pretty chubby. And this is the one that has extra of this gene. It's gone lean. And this was done in regular food or high-fat diet. And in both cases, you see that, there's a plastic response according to the diet, but you also see that the mouse that has an overexpression of this gene is leaner. So one of the things about having these promoters and the transcripts that they direct is that you can clone 5' to that promoter. You can clone a piece of DNA, and in that information, in that non-coding region, is the secret to where those transcripts are going to be put. And so Aaron cloned 5 to 8 kb of the DNA from each of those 5' to each of these promoters, and he hooked that up to a reporter type system. And we asked, well, in the nervous system, where do the transcripts go in the larva from promoter 1? And this is a larval brain, which is 2 brain lobes and a ventral cord, and it was all neuronal. And then promoter 2 was a midline glia. Promoter 3 was sheaths that wrap the nervous system that are glia and other sheaths, so perineural glia. And then promoter 4 was some neurons and some optic lobes. And this is just the nervous system. So within the nervous system, there's modular structure. Each of these promoters are putting the transcripts in different places. If you do the same thing for the gut and the fat and other tissues, you find that they're also modular. And he also did this with the adult fly, looking at sex differences in expression. And I'm going to mostly concentrate on the larva, but I'll tell you a little more as we go. So for the larva, this was the kind of result he had for those pieces of DNA that flanked the promoter. He also did a nested series of them so he could narrow down where the cis-regulatory elements may be. And remember, within this piece of DNA, there's SNPs, right? And so between rover and sitter. So we could associate, if we found differences in expression, we could associate a polymorphism in that region for that phenotype associated with that promoter. So anyway, so what Aaron did, and Ina was involved in this as well, is showed that path length, that movement in a dish, was directed by the first promoter, and likely in neurons. We did not find any phenotypes to do with the second promoter in larvae. The third promoter was all about fat, triglycerides, and then the fourth was about larval food intake. And there's another story about gut that is an isoform story where we've identified all the protein isoforms and manipulated in a subset of gut cells, showing that it's responsible for plastic responses to food intake in the gut and how much fat the gut has, but I'm not going to tell you about that today. It's not that we're only concentrating on the nervous system. And just for the adult brain, we have ideas of it. We have knowledge of where the expression is. And for the adult brain, you can also look at single-cell seq data, which Aaron Allen did, and found that there's a lot of expression in glia. So this is a brain single-cell seq data for the adult fly. And if you look at repo, the glial marker, foraging, that's the dark black dots, is highly expressed in glial cells. And that made sense to us because we knew from our synaptic plasticity studies that there's a glial phenotype. Okay, so this is a nice photo that Ina made for a review that she wrote for annual reviews of genetics. And it gives you an idea of what we know so far about the pleiotropy. And I'm not going to talk about everything today, but promoter 1, I told you, is influencing path length. There's a very nice story on pain, nociception, that is also tied to promoter 1, done by a former postdoc, Jeff Dawson. and then promoter 3 is fat, and then promoter 4 in the adult is food intake, and then in the larva it's feeding, and then there's been regulators that have been studied, also in Ina's thesis, about G9A, which is a histone methyltransferase that I'm going to talk to you about, and Pamilio, a post-translational modifier of RNA. And these are the more complicated phenotypes: sleep, learning and memory, social interactions. And so we thought we would start with these food-related phenotypes first. And with Mona, we have a project on a microRNA that is regulating sleep. And hopefully we'll be able to talk about that at another meeting. And I should say that up till now, we didn't do both sexes. We would kind of look to see, where do we find the biggest difference? And wherever we found a big difference to do the genetics, we thought, okay, we'll do it in mated females because they have the biggest difference. And then if we found a big difference in the male for the phenotype we were looking at, we would do it in males. But now there is a little bit of sex difference data, partly because I was coming here, and because, of course, it's very interesting. But just to show you that the gene is responsive to many environments, and here's a social environment. So with my colleague Joel Levine, you can put flies in a dish, and he figures out social network structure. So how information flows through the group of flies there. And one of the measures, behavioral measures, is interaction duration. How long do they interact for? And we knew that sitters as adults could only learn and remember when it had its friends around. If it was isolated, it never could learn and remember. But rovers could learn and remember whether it was alone or in a group. So we had some indication that there were differences in social learning. And so this is just how long do they interact. Sitters interact for longer than rovers. This is just a genetic background control, a rover on a sitter genetic background. And then we isolated the rover and sitter flies before they came out of their pupae, and they never had any experience with any other flies. And we saw that it made no difference whatsoever to a rover, whether it had no experience, but a sitter that was isolated, it took way longer to start interacting with its buddies. So these are all traits that we can start to do the molecular analysis on. And I just got a grant for that, so we're going to delve into these more complicated behaviors. But let me tell you about the epigenetic side of this and Ina's PhD work. And so there's Ina, you know her, she just gave a great talk. And so she had a number of projects. One of them is not published yet, and I'll go through that quickly. Pamilio RNA binding protein, and the other is about the histone methyltransferase I told you about. So this is, the foraging gene has 3' UTRs that harbor 4 Pamilio binding sites. And Pamilio is an RNA, messenger RNA binding protein. And so here's the gene, and then at the 3' UTR, she's blown it up here, and these are the normal Pamilio binding sites, and she made a CRISPR mutant that didn't knock them out, but changed the DNA sequence from T to C so that Pamilio could not bind. So that was the manipulation. And she showed that Pamilio directly binds foraging in this pull-down assay where we have foraging and Pamilio binding, a positive control and a negative control. And when she looked at the larva, the whole larva, she found no difference between the normal wild-type fly and the Pamilio CRISPR mutant. But when she cut up the animal and looked only at the nervous system, she could see that, as expected, the CRISPR mutant, there was an increase in expression in protein here for in the nervous system. And when she broke it down to what promoter is important, she found that it was promoter 1. So in terms of wild type and the CRISPR mutant, none of the other 3 were different in RNA expression, but promoter 1 was, you got more foraging, and that usually makes more of a rover, because sitters have less in their nervous system than rovers. And you can see that here, if you dissect the nervous system of larvae, there's no difference in expression in these promoters, but promoter 1, we see rover has more than sitter. And so it was the Pamilio mutant was generating the gene expression that was the difference between rover and sitter. And when she looked at what that mutant did to path length, it made it more rover. It increased the path length. And so she's done other work to ask, does Promelio affect other metabolic phenotypes and also pain? But I thought I would show you these results today. And now for the bigger story, which is, and a completed story, which is epigenetics and individual differences in adult foraging behavior. And that's the last story that I'll tell you. And just to remind you, and we've been talking about this, there's 4 different kinds of epigenetic modifications. This is a review, a primer in PNAS, and it's in a volume on the biological embedding of experience in 2020. And some of you have been citing papers from it. So it's nice for me to see that. But anyways, so in flies, we focused on histone modifications. And this is what we did. But of course, in other organisms, there's DNA modifications, and we've been hearing about non-coding RNAs and microRNAs, and then Ina gave us a glimpse into RNA modifications. So when we started this project, we knew a little bit about G9A. It's a euchromatin histone methyltransferase, a family of evolutionary conserved proteins that writes part of the epigenetic code through methylation of histone 3 at lysine 9. It leaves a repressive mark and is a key epigenetic regulator of neuronal genes. Mutations in it alter larval foraging behavior. It's also called EHMT in humans, and it influences intellectual disability. I've got allergies, so it's not COVID, don't worry if you hear me hacking. Okay, so when we looked at which of these promoters is methylated by G9A, they all were. And so we asked the question, does epigenetic modification account for the rover and sitter behavioral difference? Is it because of that epigenetic regulation or differences in it and the genetics, because the difference had been mapped to the DNA sequence genetically, are they somehow involved in generating rover-sitter differences? We studied this in mated females. It was the sex and the condition where we saw the biggest differences, but I'll talk to you about some sex differences towards the end that are not published. So this is a fly foraging arena that I told you about when I was telling you about the human data. There's a dish, the fly comes up from the middle, pokes its head around, walks up, the platform closes, and it gets to walk around the dish. There's a water moat and 4 sucrose drops. And we're measuring the path it takes and how many drops it eats. And we're interested in the movement, but also the drops. Then you saw those already. And so what Ina did is we got a deletion of G9A. So we genetically, thanks, I brought some. We genetically got rid of G9A. And so here's a G9A null, here's a G9A wild type, and then the dark is rover and the light is sitter. And so it happens in adults that rovers, as you know, eat more. They find more drops and eat them than sitters do, and that you can see here. But when we have rover and sitter and we take away all their G9A, we delete the G9A gene in rover and sitter, that difference in the number of drops they find goes away. So that was the first indication that G9A may have something to do with this because there's an interaction. And this didn't matter how long we measured it in the dish. We saw this pattern whether it was in the dish for 5 minutes or 10 minutes. And we knew because of the genetics that Ina did to make these strains that there was no difference in the G9A gene when it was wild type because the X chromosomes were the same. And also they had no difference in RNA. Okay, so then the question was, we have G9A involved. Is it methylating rovers and sitters differently? What's happening to gene expression? And the first question is, can we tie this difference to one of the promoters? And so Ina did gene expression and also methylation measures for G9A for the 4 promoters. And we were looking for this pattern where rovers and sitters were different, but when we took away G9A in the null, they were no longer different. They were the same. And we found in terms of the methylation that promoter 4 here had a very similar pattern. The rovers had higher methylation than the sitters did, and the difference disappeared when there was null. And we didn't see a difference in expression in the whole fly, but when she cut up the fly, you can do these Dracula experiments in flies and not worry about it. She dissected the brain, the gut, the ovaries, and the carcass. She saw that the brain and the ovaries had a difference in expression in the direction that you would expect. So rovers have more of the methylase and lower expression. This is expression, and sitters have less, and the difference went away in the null. So both for the brain and the ovaries, we saw this pattern of the rover-sitter difference in methylation by this histone and expression in both the brain and the ovaries. And this appeared to be from promoter 4. But we didn't want to leave it there as an association, because in flies you can really do a causal experiment. And so what she did is she used RNAi to interfere with only the transcripts from that 4th promoter. So she reduced the RNA transcripts from promoter 4 using RNA interference in sitters. And what she got was a sitter fly that behaved like a rover. It went and ventured into the food drops. So this is a number of drops. This is the proportion of time it was spent in the arena. And then this is the treatment here. These are the controls. And you can see that the manipulation of reducing or getting rid of only the 4th transcripts is turned a sitter into a rover. So that was proof that that subset of transcripts are important. And then she went to ask, well, we know there's also a genetic effect. What could it be? And so she looked 5' to this promoter and found a SNP that is in a MAD binding site. And so we think that that SNP is interacting with the epigenetic modification. So the rover-sitter SNP is a half a kb upstream from promoter 4, and we think it's a putative transcription binding site. So since that time, another postdoc, who's now a prof, Maria Aristobal, looked at the levels, RNA levels of MAD in males and females, because she's interested in sex differences, and saw that females have higher levels than males. And she also looked at G9A in females and males and saw significant differences. And then she asked the question, which promoters are important for sex differences? And it turns out that promoter 2 and 4 differ between rover and sitter and are the ones that could be candidates for sex differences in any trait that we're studying. In the case of promoter 2, the male sitter has higher RNA levels, and in the case of promoter 4, the female sitter has higher RNA levels. So this will be something we can use to expand these experiments into sex differences. So what I've tried to show you today is that it's the gene structure that is facilitating this pleiotropy, and that you can look at it in a modular way in terms of which subset of transcripts and protein isoforms, where and when genes might get expressed, and then each of them will have their own regulators. And we have also done large-scale RNA-seq of gut and fat and brain, and we see some interesting patterns in response to food deprivation between rovers and sitters. So we're doing this also on a bigger level. But before I sign off, I wanted to just show you this figure, which is about time scales, and I think it's relevant to this audience, and that really when we're talking, we're thinking about different kinds of time scales. And so the neurobiological time scales at the synapse or at critical periods are milliseconds to seconds. The gene expression time scales are minutes to hours, and we can have epigenetic mechanisms acting on critical periods and also gene expression. There's circadian rhythms, days, and developmental critical periods can be over years. And then we have maternal stress, where you have the fetus, you know all this, we talked about this, where you could have an intergenerational time scale, and then presumably a transgenerational time scale. And the genetic change is a DNA sequence which is influenced or evolving according to natural selection. Now, one of the interesting things about finding genetic by epigenetic interactions, and this is happening more and more, is it provides a means for epigenetics to be involved in the conversation around natural selection, right? So people who are hardcore Darwinian evolutionists, I have many in my department, do not like epigenetics because it sounds like a Lamarck to them. And then there's people who have the plasticity first hypothesis, where the plasticity gets you into an environment, and then maybe there's a mutation that gets selected. But if there are interactions and there's more and more data between a SNP and the epigenetic modification, then no one can argue against epigenetics having some role in natural selection. So I think that's going to become like a good discussion in the future. So to sum up, I have no idea what the time is. This is my last thing I'm saying. I can't see the time. Anyways, here's some insights. So genetic predispositions or DNA sequence variation does not preclude plasticity. The extent of plasticity for a given genetic variant is context and experience dependent. So you don't have one individual that's always sensitive to all the environmental contexts and one that's more resilient. It depends on the context. Individual differences and individual variability, which I didn't talk about, are important for understanding stability and flexibility of behavioral phenotypes. Pleiotropy, genes that affect behavior are most often pleiotropic, and the modular structure of melanogastrogene facilitates its pleiotropic functions. You may think that genes aren't pleiotropic, but everyone has picked their favorite gene and studied it, right? So you have a gene and, oh, that's to do with axis polarity in the embryo, and you think that's all it does, but no one studied it at any other time scale. And this pleiotropy is important when you think of side effects, right? So we may be interested in making a drug for something, but that something might have a function in the heart, so let's say, as well. And so you get what we call a side effect, but it's partly to do with pleiotropy. So 4 has 4 promoters that drive expression of a subset of transcripts and a subset of tissues at different times. Different gene regulatory partners and by extension networks are associated with each promoter module, set of transcripts, and the regulation of a given behavioral phenotype. And then finally, I taught you a little bit about time scales. So thank you very much.